Hi, everybody. Welcome to Chapter 3, Neuroscience and Behavior. I think I'll start this one just reminding you all that these chapters themselves are typically their own standalone course that you can take as second or third year courses. This one in particular would be much similar to Psych 2606, Brain and Behavior, or really your introduction to neuroscience. So, with that in mind, we'll go through things as reasonably slow as we can, given that it's me just talking to myself. But I would strongly encourage you to read the book, review the PowerPoint, which is available for you on the course CMS on Brightspace, and then to listen along. And of course, pause or feel free to email me, which is best to get a hold of me to ask questions about anything you may want to know more about. So, with that said, let's begin. I'm also going to do the format this way because I didn't know that the previous part 2 of chapter 2 was cut off. I'm not sure why that happened, but hopefully this rectifies the problems that we experienced. So, with that in mind, let's continue. So, the outline of the course will basically be neurons. We have the origin of behavior, the electrochemical actions of neurons, information processing. We'll discuss the organization of the nervous system, structure of the brain, genes, epigenetics, and the environment. And we'll end off with really investigating the brain. How do we look inside? I'm probably going to break this up into 2 different recordings because otherwise I think it might be too long, but we'll see. I think it's important to start with really what's critical about any behavior, especially as it pertains to humans, is that the neuron is the basis of all of the behavior, all of the things that we experience, that we feel. This is the foundation of those responses. A neuron itself is a cell in our nervous system, and it communicates with other ones, other neurons, passing information along, forming these vast and extremely complex networks. Initially, there was much debate about how behavior occurs, especially in a brain. We have staining techniques like the one you can see in front of you, the Golgi stain, that allow us to see neurons under a microscope. And when we do, we can see those unique properties of them. Ultimately, though, they don't touch each other. They are independent, if you will, and they are critical for relaying information that ultimately gets processed to become the things we talked about: our thoughts, our feelings, our behaviors. The neuron is composed of a cell body. This coordinates information processing tasks and keeps the cell alive. We have dendrites. Dendrites receive information from other neurons and they'll relay it, it says to the cell body, but they'll relay it through the neuron, through the axon specifically. We'll keep it simple, but the complex piece is that some of these connections to other neurons go directly to axons or to cell bodies, as well as to dendrites. But for this case, we're just going to say it receives information from other neurons and it relays it through the cell body. It's important to also remember that the process of information flow is always from dendrites down to the synapse, this junction or region between the axon of one neuron and a dendrite or cell body of another neuron. It flows in this particular direction and not backwards. So it never back propagates. The synapse itself is just a space between a pre and postsynaptic neuron. So the presynaptic neuron is the cell that's going to transmit the information to then a postsynaptic neuron, which receives stimuli and may or may not cause a neuron to fire. We'll discuss more about how they fire in a second. The axon is just the length of the neuron, which we'll show in an image in a second, where information transmits towards the synapse itself. And as a result of the information receiving from the dendrites, passing down through the cell body, through the axon, to the terminal buttons, so the area we call the synapse, which then releases neurotransmitters. These are really the structures that allow these cells to communicate to other neurons, to muscles, or to glands. Myelin sheath is the fatty insular layer that covered the axons. And there's a whole bunch of other support cells called glial cells that are found in the nervous system, specifically in the brain. Oligodendrocytes, astrocytes are some which form, for example, just other supportive structures, the blood-brain barrier. That's what astrocytes form. But you can see here that we have a presynaptic neuron and a postsynaptic neuron. So effectively, a neural impulse will transmit itself down through the axon to the terminal buttons or the synapse of a neuron releasing neurotransmitters that alters the resting potential then of this postsynaptic or another neuron. It's better to almost think of this simplistically as domino chains. We need a first domino to fall over before the second one will. And if we create a long line of dominoes on the floor, that's what effectively is one way to think about how neurons are sending their signals through a network. So it's not just one neuron and we get a behavior, it's a connection of how they fire together or wire together to fire together to form the behaviors that we all experience. So it is nowhere near as simple as a domino chain. It's not a serial order, which is the way I've described it. There's really a whole host of action potentials that occur in parallel or parallel processing. But simplistically put, this gives you a visualization of dendrites, the tree branches that you can see receiving information. We have a cell body, the axon covered in myelin sheath. And at the end, we have the synapse, really where the terminal buttons are, that release these vesicles that contain neurotransmitters that may or may not cause another neuron to fire. Now, I know we can't really have a discussion about what you think occurs if things don't work normally, which is where I would do this in class. But the myelin sheath is a great question that I would pose students. What do you think happens if that fatty substance just disappears? And while you may not know, I think you may be aware of what actually happens when that occurs. And that's multiple sclerosis. MS is a result of this myelin decomposing or breaking down, which causes the signals to lose their, say, efficiency. They're not as smooth, they're much slower, resulting in the problems that people who have MS present with. Neurons are also very specialized in function. There are three major types, each performing a distinct function in itself. One are sensory neurons. Their role is to receive information from the external world and convey this information to our brains via the spinal cord. You can think if you reach out and touch something, the temperature of it is cold, like a cold glass of water. That's as a result, our perception of cold is our brain's interpretation of the signals from our hands sent to our spinal cord to our brain to be processed. Much like vision, when you use our eyes to look at the screen, we're talking about light reflecting off the monitor, transmitting itself into our eyes, where our eyes relay the information to our brains in really a usable format so that our brains can interpret what the signals of those environmental stimuli are, in the case the words on the screen. And ultimately, this is true for all of the other senses as well. So think of it as our sensory neurons are relaying information from the senses. Now, I already asked, why do you think you have a brain? And that Daniel Wolpert argues we have one to move, which I agree with. It's a compelling argument. But motor neurons carry signals from the spinal cord to the muscles that result in the ability to produce movements. So think as you learn to play an instrument, your movements become more refined, making fewer errors as we do this. As a consequence, we should be able to see a different ability of our brain to fire more effectively to produce smoother motor movements. Lastly, we also have interneurons that connect sensory neurons, motor neurons, or other interneurons together. Some kinds of neurons we see, especially in the cerebellum, are Purkinje cells. In this case, you can see how the axon itself is relatively simple, but look at the dendritic branch. It's in gold, yellow. Some of these neurons themselves can have up to 10,000 connections with other neurons. You can imagine how hard it would be to trace the pattern of activity through the cerebellum if one of them has 10,000 connections, and then those 10,000 have 10,000 more. It exponentially becomes impossible to track the flow of information, especially in the absence of techniques available to do so. Hippocampal pyramidal cells are also presented just for you to look at. Just these are different kinds of dense dendritic branches to slightly more or less complex, and then to really straightforward neurons like bipolar neurons, which are found in the back of your eye. How neurons communicate are via electrical and chemical responses or reactions. So in this case, what we see is conduction that occurs in the beginning, which is the movement of electrical signals within the neurons. And to transmit information, this electrical signal has to propagate down the axon, which results in a chemical reaction or neurotransmitters being released across the synapse. Thus, together, this is really an electrical or electrochemical action, which results in the change or pattern of firing of neurons in our brains. The signals themselves, while I know it's odd to think of an electrical chemical reaction, that's effectively what it is. Neurons at rest have a charge, and that charge is about minus 70 millivolts. And as the information, or as the cell depolarizes, becomes more positive or less negative, we can see that there gets a point, there's a threshold that can be crossed. And if it does cross, what we have is an action potential. So the electrical signal that's conducted along the neuron's axon travels to the synapse. This action potential occurs in an all or none phenomenon. Imagine you're trying to flush a toilet. If you grab the handle of it, you could probably wiggle it just a little bit and initiate a slight drainage from the reservoir. But it's going to get to the point where once you push the handle, it's going to flush. That's sort of this all or none action. Similarly, once it's flushing, you can push that handle all you want, and no further flushing will occur. That's called a refractory period. So when a neuron fires, it becomes depolarized. So at rest, it's minus 70 millivolts. That's its resting charge or resting potential. If it becomes depolarized, so less negative or more positive, it may fire. And if it does fire, it's going to end up in the result of releasing neurotransmitters. And while this process is occurring, we also get this rest period where no further stimulation will result in an action potential being generated. Now, at rest, how do we know the resting potential is negative? Biologists Alan Hodgkin and Andrew Huxley discovered this resting potential in the summer of 1939 while studying sea creatures that lack spines, such as clams, squids, and even lobsters. Now, the reason that we could do this with squids, as an example, is because their axons in the squid's body is 100 times larger than the biggest axons in humans, which makes it a lot easier to study, to see, and to then test. So if you insert a thin wire into the squid axon, what you'll notice is that inside and outside of the cell, they have charges. And the difference between them, that resting potential, is with minus 70 millivolts, which is probably easier to see here. And if you think of that analogy of wiggling the handle on a toilet, you can see how some of the water escapes, but just because it's a little bit more positive doesn't mean that a neuron's going to fire, just like the toilet won't flush. But it will get to the point where, let's just say, the threshold has been exceeded. The toilet is now going to flush, or in this case, the neuron's going to fire. So at rest, in the resting state of these uh, neurons, what we have is sodium is more outside of the cell than inside of the cell, and there's more potassium inside. But once a cell fires, sodium rushes into the cell, whereas potassium's booted out briefly, which causes it to then become more positive or depolarize and to fire. And this process occurs actively with the sodium potassium pump, as well as other gated channels. And this occurs very, very briefly in about a millisecond. So we can see how it becomes more positive. The cell rapidly depolarizes or becomes more positive. And then at the bottom, you can see this refractory period where it becomes more negative than it is at rest before returning to resting potential. And all of this occurs because of the interplay of or exchange of ions that occurs in and outside of the cell. The action potential occurs due to changes in the axon's membrane channels. So at rest, the membrane channels for sodium ions are closed. When the electrical charge across the cell membrane reaches its threshold, though, sodium channels in the cell membrane open, and sodium ions rush in almost instantaneously, causing local electric charges to surge. This creates that domino effect where once the cell starts that process of firing, the electrical signal propagates down the axon into the buttons to release the neurotransmitters via vesicles into the synaptic cleft. The myelin that wraps the nodes of Ranvier help to expedite the speed at which the action potential travels down the axon. This is called saltatory conduction. In the absence of this, I believe the signal only travels somewhere around 6 meters per second. But when it is insulated with this fatty substance, myelin, the speed of signal is about 120 millimeters per second. So it's significantly faster. How neurons talk, quote unquote, to each other is via the release of neurotransmitters. So in the terminal buttons, which are these knob-like structures that branch out from the axon, the neurotransmitters are released. And these are very specific in terms of each neuron and what they produce and what they release, as well as in the pathways that are formed. So these chemicals, hence the electrochemical reaction, transmit information across the synapse to another neuron's dendrite, that postsynaptic neuron. The way to simplistically describe the function of neurons firing, it's to either influence the likelihood or lack thereof of the postsynaptic neuron firing itself. So just because a neuron fires, it's either excitatory or inhibitory communication in nature, meaning it's going to increase the likelihood that a neuron will fire or decrease the likelihood that the neuron will fire. That's it. So simply put, when neurons transmit information, it's to either result in the increased likelihood a neuron will fire or not. The receptors themselves are parts of membranes that receive the neurotransmitter and initiate or prevent a new electric signal. The receptors themselves fit like a lock and key, meaning dopamine neurotransmitters do not fit in serotonin receiving ports and vice versa. So a dopamine neurotransmitter has to fit as a key in its dopamine lock. The same would be true of serotonin. The presynaptic neuron refers to the neuron sending information, and the postsynaptic neuron is the one receiving it. Here's a better view of the terminal button of a neuron, and then the postsynaptic neuron's dendritic branch, or the receiving site. So these neurons contain in these vesicles neurotransmitters. And as an action potential travels down an axon, it results in these neurotransmitters being secreted into the synaptic gap. Now, these combined with the receptor sites to increase or decrease the likelihood that the postsynaptic neuron will fire. So, questions to consider. If a given neuron makes a few thousand synaptic connections with other neurons, what tells the dendrites and neurotransmitters to flood the synapse into the synapse to receive? So, what tells the dendrites which neurotransmitters flood into the synapse to receive them? And the question I don't expect you to be able to answer, but it's basically the pathways and how neurons fire together, wire together. So, through our development of learning how to do things, neurons effectively form these very strategic and elaborate pathways that constitutes our behavior. You may also want to consider, well, what happens to the neurotransmitters that are left over out in that synapse? Do they just stay there forever? And no. In fact, the presynaptic neuron is able to reuptake, reuse the neurotransmitters that are out there. Sometimes inhibitory transmitters are released, like GABA, that will stop and then take away the neurotransmitters that are there, so they get broken down and removed later as waste. There's more than 100 chemicals that play a role in transmitting information throughout our brain and body that ultimately affect our thoughts, feelings, and behavior in different ways. Some of these include acetylcholine, which is involved in several functions, including motor control. We have dopamine, which regulates your motor behavior and your motivation, pleasure, emotional arousal. Glutamate is a major excitatory neurotransmitter involved in the transmission of information throughout our brains. GABA is the major inhibitory neurotransmitter in the brain. Norepinephrine is particularly involved in heightened states of awareness, vigilance, to help us when we detect threats in the environment. Serotonin's that feel good, regulating sleep, wakefulness, feeding, and aggressive behavior, as well as endorphins. They act for pain pathways and emotional centers of our brains. You might be wondering, well, how do drugs mimic neurotransmitters? Well, drugs affecting nervous systems operate by increasing, interfering, or mimicking neurotransmitters themselves. Some of these are agonists. These are drugs that can increase the action of a neurotransmitter. Antagonists, on the other hand, are drugs that block the function of a neurotransmitter. So imagine you took heroin. I'm sure it would feel good, and that feeling, the description of feeling good, would be a result of the neurotransmitters being released and allowing us to interpret that feeling as pleasure. Now, the risk, of course, of taking something that's going to cause the neurotransmitters to continually be secreted or released could lead to, we'll get away from the problems of overdose. For this example, just think if the drug's causing the neurotransmitters to be released, then that's the agonist, to increase the action of them. Now, while there's a risk to it, naloxone, on the other hand, can block the function of a neurotransmitter. In this case, naloxone works really effectively of kicking out heroin that has bound to these receptor sites. It just boots them out, and the drugs are still present. They're not able anymore to have an effect on the receptor sites. So effectively, the high is over. Realistically, other drugs that are used for things not really commercially, to treat different behaviors of things like L-DOPA for Parkinson's disease, so synthetic dopamine. Because in individuals with Parkinson's, there's problems with the ability to produce it. Therefore, we can synthetically create it, and it can help produce the tremors that they experience as a result. Here you can see the actions of drugs as they increase the production and release of neurotransmitters, causing cells to continually to fire. But there's so many different ways of drugs as an example of interfering or assisting with neurotransmitters being released or synaptic transmission. This can occur from even the production of neurotransmitters to increase their production, to release the neurotransmitters. It can occur at the autoreceptors. It can occur in terms of the uptake, like prohibiting it. Selective serotonin reuptake inhibitors, as an example, increase the amount of serotonin left out there. There's postsynaptic receptors to influence the other cells to influence the ability of them to fire, and even within the synapse itself. There's also antagonists rather, that block the function. Drugs of things like caffeine can affect the autoreceptors, so they inhibit the release of neurotransmitters. It's really quite fascinating. Botulism is another one that can block the release of neurotransmitters as well. I believe for that one, the example is like acetylcholine you can inject. Botulinum or botulism, which is Botox. And it just inhibits the ability of acetylcholine to fire, which is why you can't make a face once injected in your forehead, for example. How our brain is organized is just referred to as a nervous system. So we're going to talk about the central nervous system and the peripheral nervous system. So we interact with these networks of neurons, and they convey electrochemical information all throughout our bodies. So the central nervous system is composed of your brain and your spinal cord. In this instance, we can talk about how our brain receives information from the senses, via the spinal cord, as we mentioned earlier, so from the external world. Our brain can process the information. It can coordinate and use it to help us move, issues commands to the skeletal or skeletal and muscular systems for action. So our brains can support the perception, our motor functions, our emotions, our cognition. And the spinal cord is really just this branch down from the brain that can execute the commands sent from it. So you have to remember too, it's all about the information coming into the brain, going up to it, as well as coming away from it. So our nervous system, you can see, if we really break it up, the central nervous system just becomes your brain and your spinal cord. And then the peripheral nervous system includes all of the autonomic and the somatic breakdown of these systems to help with our arousal, sympathetic or parasympathetic, calming effects of behavior that can occur. So the autonomic nervous system really deals with the automatic, involuntary control of internal organs and glands, and the somatic, which contains information into and out of the central nervous system, which control our voluntary movements, our skeletal muscles. The peripheral nervous system connects to the central nervous system's body organs and muscles, which we just briefly mentioned. The somatic nervous system conveys information into and out of the central nervous system. And the autonomic nervous system really carries involuntary and automatic commands that control blood vessels, body organs, and our glands, which is divided into the sympathetic nervous system, which can prepare our bodies for action, the fight or flight, as well as parasympathetic, can help the body return to its normal resting states. In the event we detect a risk of some sort that results in this heightened level of awareness, arousal, the sympathetic nervous system would result in our eyes or pupils becoming dilated. Our airways, if you will, would constrict, our heart rate would increase, dietary processes would slow down, grind to a halt. We get glucose released, we end up with epinephrine, norepinephrine, adrenaline. The bladder can relax. Basically, it prepares our body, the dry mouth you experience, things of this nature. Increased heart rate, breathing, everything basically prepares us to fight or to run. On the other hand, when we have to relax, we want to see the body return to normal, which means our heart rate has to slow down or breathing relaxes. Digestion returns. All of the other features that are basically suspended return, which is a really important life-saving, if you will, evolutionary feature. The central nervous system is comprised of the spinal cord and the brain. And I think the main distinction we talk about there are neurons and sending information in our brain and inside of our body itself is that if we damage a peripheral nerve, they can regenerate. You've probably all cut your finger, and of course it hurts, and you might notice a little bit of, I don't know if numbness is the right word, but let's just say you touch it and it just doesn't feel right. As time it heals, though, everything seems to return to normal because they regenerate. The challenge when we damage our central nervous system is that they don't. So, damage to your spinal cord, if severe, is permanent. Think of paralysis. Now, imagine we damage our brain, whether it's stroke or some other form of brain damage, your brain will not heal itself. The damage that occurs will not regrow. That is the main distinction between the two. Also, we have spinal reflexes. You've probably all grabbed something that's a little bit too warm at one point, and you'll notice that the reaction when you do so is to withdraw immediately. And that's a reflex that occurs in your spinal cord. There's no conscious processing of that information. You don't touch it and evaluate, oh, that feels pretty warm. It just is. And we just automatically pull back so we don't damage our skin. A reflexive arc is a neural pathway that controls reflex actions, like when we reach and touch something that's warm. Not that I'm going to encourage you, as the illustration suggests, to reach out and put your finger towards a flame, but if you've done that, you know that if it's too hot, the receptors immediately send signals through the spinal cord to pull our hand away and avoid further damage. The spinal cord itself can be divided into four main sections, each controlling different body parts. The cervical spine, or C-spine, is listed for you to see in the top. And C1 and 2 are looking at nerves that come out and support or send information to our head, our neck, our diaphragm, deltoid, biceps, wrist extensors, biceps. Then we get to thoracic nerves like chest muscles, abdominal muscles, to lumbar, and then to sacral nerves. Damage on the higher end of the spinal cord usually means greater impairment, the quadriplegia. Damage that occurs lower, often paraplegia, the loss of use of your legs. And if it's very, very high, you could see how you would have difficulty breathing, which is we're going to require some assistance for the remainder of your life. In the real world, the idea of a brain-computer interface isn't new. So we know that neurons transmit information based on the input it receives from other neurons, simplistically put. And that information is done via neurotransmitters as a result of depolarization or cell firing. In the real world, people have tried to train and they have successfully trained monkeys to learn to adjust the motor cortex activity to control cursors with thought. Meaning, if we understand how the motor cortex fires, we can insert electrodes in there and teach monkeys with just thinking about moving to actually move a cursor, which is pretty neat. We've also seen successfully women with tetraplegia who learn to control robotic arms using implanted motor cortex machine interfaces. And recent studies are increasing our focus on decoding an individual's goal and intentions based on signals in the frontal lobe, which is very critical to high-level cognitive functioning. The brain itself can be divided into three major divisions. One's the hindbrain. This coordinates information coming into and out of the spinal cord. It also controls the basic functions of our life. So in this instance, the medulla, the reticular formation, the pons are all at the base of the brainstem, followed by the cerebellum outside of that. The cerebellum is really critical for things like movement. And it also contains about 86% of all the neurons in your brain right there. So remember the Purkinje neurons we looked at earlier, that they're really dense dendrites? They're in your cerebellum, and there's almost all of our neurons just in the cerebellum itself. So things like breathing, heart rate, nausea, these are all, and consciousness, dealt with by the medulla, the reticular formation, the pons, and then movement through the cerebellum. The midbrain is really important for orientation and guiding our movements via the tectum, the tegmentum, as well as we have the forebrain. The thalamus should also have been mentioned in the midbrain, but let's just leave that. The forebrain is also the highest level of our brain. We talk about it this way. It's really the neocortex, cerebral cortex. It's absolutely critical for complex cognitive thoughts, emotions, or sensory and motor functions. We'll get into much more detail further in other courses, but for now, that's probably good. If you think about building a brain, which is what I ask students in class, where would you start? And it's not uncommon to think of it as building a house. We need to start with the foundation. And that foundation, when we break it down, really is our brain stem, the beginning here, where we can see information's coming in uh, via the spinal cord. And it's probably easier to visualize here. You can see the medulla, which regulates our heart rate. The pons is relaying information to the cerebellum and to the rest of the brain. The cerebellum controls fine motor skills. And the reticular formation is responsible for consciousness. If this was actually damaged, you'd be permanently unconscious. The midbrain is just sitting between the brainstem and then the hindbrain, which is your tegmentum and the tectum that's shown here. And then if we look at the actual forebrain itself, we can see a whole bunch of complex structures. The corpus callosum, which is just a division between the hemispheres, the basal ganglia, which we'll talk about that in a second, amongst all of the other areas that are here. The thalamus really is a critical subcortical structure that relays all of our sensory information to the cerebral cortex to be processed. It is part of the limbic system, and only one of our senses does not pass through here. Can you guess which one? If you guessed smell, that would be correct. Smell is the only sense that bypasses or directly gets processed by different regions and not passing through the thalamus. Now, if you're wondering, does all of the information from, say, touch and sight and hearing uh, go through the same areas of the thalamus? No. They're in adjacent areas, but they all have their own, think of it as superhighways, where the information is passing through, going to other other areas of the cerebral cortex to be processed. The hypothalamus is responsible for regulating our body's temperature, our hunger, thirst, sexual behavior. It's also part of the limbic system. The subcortical structures, like the thalamus, receive inputs from all of the major senses I told you, except smell. In fact, it goes directly to the amygdala, which can elicit emotional responses. Have you ever had that experience of, I don't know, walking through the mall and you smell something like someone's perfume, and it can take you back to an old teacher that you had, as an example, many years ago? Just in an instant, it's like you're there again. The limbic system is a group really of structures that are involved in our motivation, our emotion, and our ability to learn and critically remember. The hypothalamus, we talked about, regulates body temperature, hunger, thirst, or sexual behavior. The hippocampus is absolutely critical for the formation of new explicit, think of it as conscious awareness, memories that can integrate them into a network of knowledge so that they can be stored in definitely in other parts of the cerebral cortex. Now, when we talk about memory, we'll talk about sensory short term and long term. Yeah, long term literally just means forever, basically. We're not entirely sure of the extent of what long term really is. But the fact that we can remember memories for decades is really implying that they're stored somewhat indefinitely. But if someone says, well, aren't all experiences coded indefinitely or experience, we just didn't remember them? No, our brain's not possible. It's not possible to remember everything. That would just be overwhelming. If you think about all of the experiences we have, all the stimuli present in the environment, it'd be literally impossible to remember everything. But it is critical for the formation of new explicit memories. Now, you may be wondering, well, how do we know this? Well, back in the 1950s, a patient named HM was experiencing seizures. And as a consequence, Dr. Schofield decided to remove the area, the limbic system of HM's brain, because that was the site of which the seizures had originated. There's probably a lot more I could say on HM, which will come up later on, but effectively, the surgery was a success. He didn't have seizures anymore. But as a consequence of removing the limbic system, which included mainly the hippocampus as well as the amygdala, HM never formed another conscious memory again. Now, he was in his early 20s when he had surgery, and he died in his late 80s, I believe. But every day he woke up, you can imagine he didn't know he was in his 80s. He would have thought he was in his 20s. He's the most studied person ever in psychology. Like, it's not even close. Not that it's a competition, but unfortunately, or fortunately for not having seizures anymore, the surgery wasn't really a success then. In fact, to my recollection, I don't think it was ever performed again. While he would have been seizure free, he really did struggle to learn new things. And he could learn new things. He just didn't know how he would have learned them. It's probably a topic for a different time, but the unfortunate thing is we know how critical the hippocampus is because of him. And subsequent work using food storing birds, rats, and damaging their hippocampus after they learn to store food or to solve complex mazes, in the case of food storing birds and rats, respectively, damaging their hippocampi results in their inability to perform the tasks that they once knew. So the birds won't know where they hid their food, and the rats will not be able to solve the maze. The amygdala plays a critical role in emotions, and we know this because, well, there's at least one person that doesn't have one. And that's kind of the fascinating thing about case studies is that we're able to sometimes find the odd individual that can help us understand how a brain works, often when there's brain damage, and sometimes when structures just don't exist. Now, in the absence of an amygdala, you can guess this person had no fear whatsoever. Snakes, spiders, which usually terrify people we don't like to look at, didn't bother her. She tried to pet a bear, not a good idea, but she showed no fear at all whatsoever, emphasizing the role that the amygdala plays in our emotional processing. The basal ganglia, and I'll just make a note that this slide in particular deviates a little bit from the stock one I provided, just so you get a little bit more information. But the basal ganglia is a set of subcortical structures that are responsible um, for our intentional movements and play a role in reward processing. Now, the basal ganglia themselves are comprised of the caudate nucleus, the putamen, and the globus pallidus. Together, they all receive input from the cerebral cortex and send outputs to the motor centers of our brain in the brainstem. The striatum is comprised of the caudate nucleus and the putamen. And if you end up with damage in the putamen as a result of dopaminergic pathways breaking down, this is what we call Parkinson's disease. If you have degeneration of the striatum, that's Huntington's disease. Other areas, even within what we call the basal ganglia, are also thought to be linked to ADHD, but they are also linked to Tourette's. So Tourette's, Parkinson's disease, and Huntington's disease are all movement disorders. I also remember reading somewhat recently that when witches were burned at the stake, it was recently hypothesized that it was possible that some of these individuals had Tourette's. Because think of the uncontrollable tics that can occur, as well as the, well, foul language that can come with it. It is involuntary. And even early documented cases of Tourette's um, highlighted how some prominent people who were well educated spoke articulately, as you would expect, also used profound profanity when these tics would occur. The idea that it could have been something like possession isn't that surprising, and it makes a compelling argument as to whether witches or some of the individuals, I'm not saying all, but some had Tourette's. Of course, there could have been other reasons, but I won't digress too far. Um, this is particularly relevant as well. I mean, if we think about, especially today, using social media, how many people take photos and you post them online and you're waiting to see who will like them? And if that's ever happened to you, um, think about when your post generates a lot of likes. How does it make you feel? Conversely, how does it make you feel if your post doesn't get any likes at all? So researchers used fMRI to examine different adolescent brains while using social media. Participants received few likes in response to the posts. They reported more negative thoughts and feelings and this greater sort of depressive experience about thoughts in general and themselves. This effect was especially strong amongst adolescents who experienced peer victimization at school. This feeling of rejection among adolescents was also linked to insufficient positive feedback as opposed to explicitly negative feedback. So the reward associated with receiving likes on Instagram or Facebook is linked to the engagement of underlying reward processing regions of our brain. So it's not that surprising to think what makes us feel good is, of course, these dopaminergic pathways that become active. I think I'm going to pause the lecture series here and continue with the endocrine system in the second part. I hope you enjoyed this lecture so far, and we'll catch you at the second one soon. Thanks.